Hi everybody, welcome back to the second Live from the Lab.
Here we are holding some boxes of the samples that we will be discussing today. And while we wait for Meredith Elliott, the senior scientist of this lab, we want you to guess how many you think we've done since June of 2020.
Yeah, we have another box back there, so.
Yeah. Just let us know. We're gonna wait for Meredith to join us.
Yeah, we'll wait a couple minutes for you all to join in.
Yeah, thank you for tuning in.
Yeah, just a reminder, we're going to be having you all guess how many samples we've done.
Since June of 2020.
Yeah. These are, in each one of these dishes is one of our least Tern poop samples.
And puke. We'll be talking about what we do with these samples, where the data goes, and all that.
Let's see. Oh, it looks like Meredith's trying to join.
Alright, we're gonna add Meredith. Lishka guesses 100.
100. Okay.
Very nice. Yay!
Yay!
Welcome,
Is it
Meredith.
working?
Yeah!
Yay!
Oh no. Actually,
Okay.
it looks kind of stretched.
Oh no. Okay.
Oh no!
Is it? I have it oriented right.
Screen.
Is it not working?
No, you look pretty stretched.
Okay.
Yay.
Okay.
All right. I'm out.
Okay.
Alright.
Thanks, Meredith.
Sorry about that, everybody.
Okay.
Alright. Yeah. so
Technical
sorry about
difficulties
that.
are following us everywhere.
Yeah. So what Meredith was going to say was just an intro for the lab. So Meredith is our senior scientist, and she will also be in the comments answering questions. So look out for her. Sorry. Okay. Yeah, so basically Rebecca and I live together, so we are considered a family unit, and that's why we can work together in the lab without masks on. But as of right now, the Point Blue headquarters are closed to the public because of COVID-19, but we're looking forward to having visitors again. Yeah, so I'll get into it. Welcome back to the lab, everybody. Today we're going to be talking about our least Tern poop diet. But yeah, so the lab has been open since 2006, and we're located in the headquarters in Petaluma, California. And since we've been open, we've had about 30 marine lab assistants like Rebecca and I get hands-on experience with marine food web projects. So we look at mostly the diets of marine predators. So like the least Tern would be an example of the seabird, which is a marine predator. And then we also look at zooplankton communities during our access cruises. But today we're going to be focusing on marine predators. And we track these changes over time. So Point Blue has a long-term data set. And so we can see how changes are occurring. And also that tells us how the health of our oceans is changing over time. So with that, I'm going to turn it over to Rebecca to talk about the endangered seabird we're going to be discussing today.
Yeah, so if you guys tuned in to our first live stream, we will be talking about the same bird, which is the California least tern. And terns are a group of seabirds, and they're actually in the same family as gulls, which I'm sure most of you are familiar with. But yeah, so least terns are the smallest tern species in North America, and we will be focusing in on the California least tern, which is a West Coast subspecies of the tern group. And so this is the general shape and size, and they're small but very mighty birds. They're about the size of an American robin, if you're familiar with that. But yeah, so these birds are unfortunately endangered, and some of the threats posed on them are habitat loss, nest disturbances, and predation is a really big one. But conservation efforts that are being implemented include signage at nesting sites, fencing, habitat enhancement, as well as restrictive access on beaches where they're nesting. So as their name implies, they nest here in California along the coast, and their breeding colonies range as far as the Mexican border up north to San Francisco Bay. And so they eat fish. That is their main diet source. However, they have been known to eat shrimp and other marine invertebrates, and even sometimes insects. And these birds, the way they catch their fish, they are aerial divers. So they will fly and hover over the ocean, see a fish, and then dive after that fish. And then they will either eat this fish in flight, like that, or bring that fish back to their little chickies or their mates at the breeding colonies. And so because these birds are endangered, biologists survey many of the colonies here in California just to gather population data and see how they're doing. And while they're out there in those colonies, these biologists so kindly pick up least tern, poop, and puke for us, and they put those in little baggies and send it here to the lab for us to further analyze. And so why do we study the diet of these birds? Well, let me tell you. We not only gather information about the California least tern themselves and what they're eating and how they're doing, but these diet projects really give us an inside look into how the ocean is doing. Because these, the prey communities, the fish, they fluctuate with different conditions of the water, and because this project is a long-term project, we can look back years ago and see how that has changed over time. So yeah, just a good look into how the ocean is doing, which is very important. So I will turn it over to Olivia to talk about what we do with this poop and puke.
Yeah. So I don't know if any of you guys tuned in last month, but we were talking about the drop fish. So these are the drop fish that were dropped at these nesting sites and picked up by scientists, and then we identify them by the whole fish. But actually another way we can look at the diets of these birds is to look at their poop or their puke, so their pellets or their feces. So yeah, as Rebecca mentioned, they come to us in these little baggies, and they're full of sand and other things. So kind of the first step we're going to show you is to clean up these samples and to get the feces separated so we can look at that. But the other stuff we remove from the sample is, I don't know if you guys can see there, but there's kind of the white stuff that you guys probably have seen in bird poop, which is actually kind of like their pee. It's uric acid, and it usually clouds our samples, so we'll remove that before we start looking at the feces because it doesn't tell us anything about the diet. But yeah, once we put the feces into a dish with alcohol, we'll use like forceps to pry apart the poop and look for two things in particular. So we're going to look for scales and otoliths. And so these are otoliths, and you can see there. They're actually the ear bones of fish. So they're calcified structures that kind of help the fish with balance. They're located in the head of the fish right here. And each fish has a pair of these ear bones that are actually mirror images of each other. So by finding these otoliths, and they're specific to each fish, so there's certain characteristics on these otoliths that we look for that can tell us which fish species this came from. And so we can match up these otoliths and we can get kind of a rough count of like how many fish they're eating. And we'll talk more about this in our next live stream with the Brandt's cormorants. Those are pellets that contain mostly otoliths that we ID, and lots of different kinds of fish there. But yeah, and then another thing we look for are the scales. So each family of fish has kind of their own unique scales, so they look a little different. And we have this guide here that helps us determine which scales these fish came from. So yeah, and on different parts of the body there are different types of scales, but we have a whole reference collection that we can refer to help us identify which scales these came from. So And
What other things do we find?
We also take note, oh yeah, we count these scales. We count whole scales and partial scales on this clicker. And then some other things we count are vertebrae or eye lenses, and then also secondary otoliths. All of these things, we can't tell what species of fish they came from, but we can tell like that they are eating fish, so that's important to know. And then some other things that they find, like Rebecca mentioned, are insect parts, or also sometimes we find plastic in our samples. And so it's good to know which samples have that in case you want to refer back to those samples later. But yeah, with that.
Yeah, we're going to go through the whole process with you under the microscope, so it might get a little blurry as we transition over there, so just bear with us. But let us know if we need to repeat anything or if the audio is a little funky. We'll be happy to do that. We'll be keeping track of the chat box, and Meredith will be on there answering questions, and we can answer questions as we go. But yeah, and then also in the beginning we mentioned we kind of want you guys to guess how many samples we've analyzed since June of 2020. There's also a little box back there. So yeah, just ponder that as we go through, and then we'll ask for your guesses.
Yeah, and those came from 5 different colonies. So.
Yeah.
Okay.
So here we go.
So the first step is to take this baggie, and then we'll cut it open and then empty it into this dish, which is what I'll be doing as we set up the microscope.
Quinn's given his guess already.
150.
Wow,
Yes.
nice guess.
Oh yeah, Sarah asked, least terns eat insects as well? Yeah, sometimes. In especially our Southern California samples, we'll find lots of insects because the fish populations aren't doing so well down there. So
Yeah, so when we find those, it's usually not a good sign.
yeah. All right, can everybody see what we're looking at? Yeah, so
big
that's
old poop.
all. Yeah, that's poop there, the darker parts, and then the rest. This is all sand. Yeah, but we don't want that for our sample. Yeah, so the first step is we will break apart the white stuff to try to get just the feces. So kind of break that up. See how it just falls off there? So then we'll take this poop and we'll place this in a dish with alcohol to look at. Well, sometimes they break apart and we just try to get all of it, what we can. Yeah, so you guys can see well? There we go. Yeah, and then with breaking this apart, it kind of flies everywhere, so.
Yeah, our fine motor skills are on point.
Yeah. Yeah, so if you guys can see the sand and, oh, that doesn't. The sand and the uric acid we're trying to keep away from what we're looking at. All right.
Quinn asked if this is poop or puke. This
is poop. We'll show you some puke later. And that's usually clear, so it doesn't have all these dark parts on it. Okay, so now that we have the poop separated, we'll add some alcohol. Yeah, and then we'll switch the light to the backlighting so you can see the scales that we find. And then we'll zoom in. All right, how is that for everybody? See that? Okay, so then kind of poke around here. Ah, I see our first scale. It's right there. Let's see if I can get tweezers. Yeah. Looks like we got some scales right here. So these are ones we commonly find. I don't know if you guys remember from the last live stream, the most common fish we get are jack smelt or top smelt. And these scales here are from those fish. And so we can identify them by a couple different things. So mostly we look at shape. So these, it's kind of hard to tell, but they have like squared off edges at the top. And that's really indicative of Anthenopsid, the family Anthenopsid, which has that top smelt and jack smelt. And then here we can see a vertebrae. So this is the vertebrae of one of these fish that they ate. Yeah, and so usually when they're hardened like this, we count them as vertebrae, but then sometimes we'll find softer vertebrae, and those are from larval fish. It hasn't quite hardened like this one has. So, and then those are considered their own family of fish, or the fish larvae, because they usually don't develop scales until they're older. So we just call those fish larvae. Oh, here's a better view of that scale. Yeah, so you can see here that they're pretty squared off. So there's 90-degree angles kind of at the top. And also we look at other things like the rings around it are very distinctive for each different fish. So these ones, it's kind of between the rings, it's kind of like wider. Some of them will find these little lines are really close together. But yeah, each one of these scales is like a fingerprint, and we can identify what species from these characteristics. And then some other types of scales will just have like straight lines across, and it won't have that circle in the center. That's a different family of fish. And then other ones could have kind of lines that go through the scale, and that could be like rockfish, or some other fish have that, but these scales here don't. So, but yeah, these are pretty common, these Anthenopsid scales. Yeah, we can keep looking around. So yeah, we would go and we would count these scales individually.
Yeah, and we also count the partial ones. So even little itty bitty parts of those scales that we can identify, we also count those, because those can give us a count of how many of those scales and how abundant that species is within this sample.
Yeah, see, this one's kind of covered in that uric acid. So if you kind of poke around, you can start to clear it up and see more characteristics of that scale. Oh, can I ask a question?
What magnification are we looking at? Just wondering about how big these scales are.
This is at 40.
40 times
what you would see.
They're very
small. This needle is, yeah, it's a needle, so if you can imagine how small that is.
Wondering about the scale of these scales. Yeah, they're pretty tiny. These terns eat small fish, but we also look at the diet of California sea lions, and those are bigger scales because they're eating bigger fish. But these least terns are eating smaller fish, so we definitely have to use the microscope in order to identify them.
Yeah. Looks like we're finding mostly Anthenopsid in here.
Shell said, when a bird eats plastic, can you tell by the color of the plastic / how hard is it to classify the piece as plastic?
Oh.
We definitely see color. We know that, we get a lot of blue, which is strange, blue fibers, and they kind of look like hairs, and they're pretty obvious because like we're just finding scales and like miscellaneous fish bones, but then once we see that fiber, it's pretty clear that is plastic. We can't really go down to the composition of the plastic, like the polymers and stuff, but we just note the color, what kind of plastic, like fiber, and how many.
Yeah, it looks like mostly Anthenopsid scales. Is it? Oh.
maybe we can switch over to
Yeah.
other scales that we find.
Yeah, so you guys saw our most common one, but we'll show you some other ones that we'll get.
So these are slides that are prepared for us to reference when we are unsure about what we're finding. So here we got some mosquito fish. See that focus is way bigger than the anthenopsid scales.
Sarah asks, are the scales pretty hardy? You don't find yourself worrying about tearing them when you're taking apart the sample. Yeah, actually that's another characteristic we look for when we're identifying scales. Some are definitely hardier than others. So the one you can see right now is pretty flimsy, and these tend to fall apart. This is from northern anchovy. And usually when we get these samples, it's full of little bits of this type of scale. And usually, since the scales we're counting don't tell us like how many fish they're eating, it just tells us the presence or absence of a certain species. It doesn't really matter if we tear them apart, but yeah, we make note of like how many partial and how many whole scales we're finding.
Yeah, good question. So that was northern anchovy. And then these are going to be rockfish. It's a terrible one.
Yeah, these you can see they have more of those lines that run kind of through the center of it. And those radii can tell us that it's a rockfish. And then also, we can find a good scale.
Ah, here we go.
Yeah, you can see right there in the center one, kind of how it goes towards a V at the bottom, and that's very indicative of rockfish. And then something you can't see in this picture is what we call teni. So they're like little teeth that attach to the fish's body. And those sometimes will be found on rockfish scales. Some fish don't have those.
Right here.
Yeah, they would be there. You can't see them in this slide, but yeah, they might have like these tenoid parts to them. Oh, you can kind of see a couple on that one.
Oh yeah.
Yeah. And so if we find the tenoid scales and we're kind of between two species, we can check to see if they should or shouldn't have these little teeth at the bottom. Yeah.
Yeah. And then this is Pacific Sari. Ooh. These are really cool because they range on the body of the fish. Like,
yeah, pretty drastic.
These two are from the same fish, these two scales. And you can see one has really fine lines that are parallel to each other, kind of like have this opening in the middle. And then this one is more circular. And even this part right here sometimes has a blue tint to it, or like a green tint. And that is really cool. Pacific Sari.
Yeah, this is one of our other most common species that we find. Yeah, and their scales, for me, I think they're the easiest to identify because they usually stay whole like this, and they're usually pretty round. So they don't have any of those tenoid teeth on the bottom. Yeah.
And then we could do one more.
Oh.
Surf perch. These are really cool too. And those dots on those scales are actually from the scales too.
Yeah, so Lishka asks, are there any similarities between the design of a scale and the design of otoliths within the species?
Oh,
huh.
It's interesting.
I haven't noticed anything. I know the more I look at these scales and otoliths, I like see their shapes everywhere in my life that aren't even like scales or otoliths. But yeah, I think definitely like rockfish, they tend to like be more like hardy, and their scales are usually like thicker, and like their otoliths are usually like
Very
thicker.
thick.
Whereas northern anchovy, their otoliths are really thin comparatively, and also their scales are pretty flimsy. Yeah.
So yeah, there are some similarities.
I guess so, yeah. I've never thought about that. Good one, Lishka. Marissa asks, can you tell which specific species of rockfish the scale is from? No.
Not really.
They all, we generally group them to the family. So this will all be Scorpionidae, which is rockfish. Even with the otoliths, because these are usually really small fish that we're finding, they don't develop those defining characteristics that we can get to each individual species of rockfish, because I'm sure you know from all your marine science classes, they are very similar looking, and so are their otoliths and scales.
Yeah, so what we have now under the microscope is a pellet, broken up pellet. So this is the puke. It's really easy to identify things within pellets because it's clean and everything is really intact, unlike poop that has gone through the digestive system. So yeah, we got some anthenopsid scales going on. We have lots of vertebrae, which we also count, and just miscellaneous fish bones.
Sarah says scales look a lot like fingerprints. Yeah, that's what I think. And they are very specific to each fish, which I find really cool.
Yeah, super interesting.
Yeah. And then Sarah also says, how many species can be found in one sample? Does it mean it ate many different species within one day, perhaps? Yeah. So some of these samples only have, like, say, anthenopsid scales, but then others will have many different kinds of fish. Depends on where they're foraging, and also depends on the colony we're looking at. It depends on what prey is available to them. So I think I found some samples that have like 4 or 5 different species in it. And usually that's a combination of different otoliths and different scales that we find.
Speaking of otoliths, should we show some? So these are the inner ear bones of fish. They help orient the fish, help with balance. There we go.
Yeah, so these are little tiny otoliths. These are only a couple millimeters long, if that.
Yeah, we are at Magnification 4. 40.
40 times.
40 times what you would see. And you guessed it, these are anthenopsid otoliths, because that is what we find most often. And they're pretty cool. They got nice little scalping edges to them. Got a little nose. But yeah, as Olivia said, these are just calcified little structures, and these you can easily break with forceps. So this is what we have to be really careful with when we are transporting them out of the sample.
Yeah, and we'll look for things on here like the sulcus is very distinct between species. So that line that runs kind of along the center, if you can see in that otolith there, there's kind of a groove that runs through there. And different species will have, like, some have a pretty shallow sulcus, some it's really deep and defined. Some curve up at the end, kind of like this one. And then some just go straight across. So the sulcus is usually what I find most helpful. And then also the general shape of the otoliths. But since, remember, these are going through the digestive tract of a bird, they tend to get damaged and broken. You can see some of these are chipped.
Yeah, they're not always
perfect
condition, which makes it a challenge for us to identify.
Yeah, but we also have a large reference collection of otoliths to refer to.
Yeah, these are very small little guys.
Yeah.
And yeah, so that is our time on the microscope. We'll switch over to answer some questions that you may have.
Looks like Sarah has a question. Do scales give any nutrients to the birds? And our senior scientist, Meredith, said, I don't think so.
No.
Yeah, I think they're made of keratin, which is like the same stuff our nails are made out of. And that usually doesn't have too many nutrients in it.
Yeah, that's why they're deposited
in the feces.
Yeah.
like I said, sometimes we don't find scales, we just find really tiny otoliths. So any otoliths that are under 0.5 millimeter, which is really small, we consider those from larval fish. And those won't develop scales, so we won't find any in those samples. So we classify all larval fish together.
It's one family.
Yeah, it's one family. So
yeah. You want to show them where we put the otoliths?
Oh, yeah. Yeah, since they're so tiny, you can't even see them with your naked eye. They're about the size of like a grain of sand, maybe. Yeah. Sometimes they're smaller. We put them in little capsules. These are pill capsules, actually.
And we don't want them sitting in the alcohol because we do store every sample that we analyze. And we take them out of the alcohol so they don't degrade in case we want to go back and look at them.
Yeah, so we use like the little capsules and put them in. It's really cute. And we also put a little strip of paper in here with the sample number so we can go back to them.
Yeah, you want to show a data sheet? So yeah, as we go through the sample, like Olivia mentioned, we're counting each species, whole and partial scales. And then we record that on a data sheet that looks like this. We give every sample their own sample number. We record where it's from, like which colony we retrieved it from, what date they collected it, and then what we find.
Yeah.
And how many.
Yeah, that's really important for when we go back through the data and we're trying to run the analyses. We can see if there's any we need to like go back and check on, see if we did it right. But,
yeah, exactly.
It's really important.
Let's look at these questions.
Okay. So Lishka says, what are some of the bigger conservation challenges that data like this can help figure out and address?
Yeah, well, observing fish communities is pretty difficult just by us observing. So by collecting data about what these seabirds are eating is they're like our little scientists, our little samplers going out there and sampling the ocean because they know what's best for them and they know what they want. And then if that is not available, they resort to something else. So we can kind of see what is available within each year, within each season, and how much and what has gone out of their diet since the other years. So we can just see how prey communities of fish are fluctuating throughout the years, which in turn tells us how the ocean is doing as a whole with different water conditions and such.
Yeah, and I think it's really important to couple that with the scientists collecting breeding data out at these colonies. So that's like really important. So we can really track how each one of these colonies is doing relative to other colonies, and then we can see the breeding success, and if a certain fish species is present or absent from the diet, we can maybe make connections there.
Yeah, for example, the northern breeding colonies in Northern California are doing pretty well, and we see that in our samples. We get a lot of scales and a lot of vertebrae. However, the colonies in Southern California, we get a lot of insects and just fish larvae and not a lot of scales, so it's kind of like these birds are resorting to insects when they should be eating fish. So it's kind of like what's going on in the waters down south where really good fish is not available to them during the breeding season.
Yeah, and that's like super important, right, for the health of the whole colony.
Yeah. Good question.
Yeah, good question. Quinn says, do all scales make it through the digestive system, or do some get broken down completely before they are discarded in feces?
He asks this because it seems like there would be a lot of scales if none of them are broken down. Yeah.
I think they are. And then also, if you notice that pellet, which is packed full of scales, whereas the poop, not so much. So kind of what I've noticed is like if it does kind of get clogged up there and they have too many scales, they just form a pellet and puke it up. And that's why we might not find some scales in their poop. But usually we tend to find scales more than not. Identifiable scales.
And sometimes they're all partial, which
Especially
is a challenge.
The anchovy, they just tend to break apart. But at least those have pretty distinct lines that run through them, so we can tell that those are anchovy. Yeah, good question, Quinn. And Lishka says, who do you share these data with? What sorts of partnerships are involved?
We share this data with those collecting breeding data out in the colonies so they can, relate it to breeding success. Mm-hmm. And then it is written up into a big report about how they did this season, what they ate, what was dominating the diet. We make a lot of graphs and just like big data on like what species was dominant, what was absent, and that just goes into the field report that is sent to biologists working with these birds.
Yeah, Meredith says we share with federal U.S. Fish and Wildlife Services and state California Department of Fish and Wildlife agencies to work to protect the species. Yeah, so I also really like how Point Blue as a whole is really collaborative with these other agencies. I think that's very important. Yeah, and it makes our work more impactful.
Yeah.
Yeah. Okay, next question. Lishka says, as an early career scientist, what has surprised or delighted you about the way of studying the ocean? Or about this way of studying the ocean?
I think it's really cool.
Yeah.
Because, I think in the past, in order to gather population data of these fish, we had to, actually catch them and then count them, whatever they did back then, which definitely has an impact. So if we can study the communities via these seabirds who are already eating them, it's a way less impactful way.
And like that method Rebecca is describing is very resource intensive. You can imagine getting boat time and sampling these areas is like a huge area to sample. But if we're really trying to focus on the fish populations and the bird populations and how they're going together, I think it's really important to use these birds as little scientists, like Rebecca was saying, to show us what is best for them. And so I think that's really cool about these projects, especially the diet work is it's so cool. And it's non-invasive, so you don't have to, kill any seabirds to look at their diet. So
it's just a way more efficient way.
I think it's really cool, and it's something I was not exposed to before this internship. So
definitely.
Okay, so Brent says, how many and where are the colonies of the samples you get?
We we get these samples from 5 different colonies in California.
And that's Point Magoo, Tijuana River Estuary,
Camp Pendleton,
there's a colony there, Alameda in Hayward. And Alameda is kind of our main site that we go out and survey, Point Blue. But the other ones, they're sent to us from these Southern Californian breeding sites. Good question.
Yes.
Okay.
Oh, Mary said 12.
Oh. Yeah. Oh, that might be a hint to how many samples we've looked at this year.
Yeah, just
Mary
a reminder
that we try to collect at least 100 samples, if possible, from 12 different colonies. Yeah, I guess within each of these colonies, there are different locations. So, I know there's different nesting sites. So within these colonies, remember there are multiple sites. So if we can get everybody's final guess on how many samples you think we've done
since June 22nd of 2020.
Yeah. Quite a few in here.
And then there's some
at math. There's stacks of these.
And then also little otoliths that we put in these pill capsules. So it's a big project. It's been running since 2002. 2002?
Yeah.
So we can really go back and see how their diet has changed throughout the years.
Michelle says 356.
Okay.
Mishka says 100.
Ash says 200.
200, yeah. Quinn changes his answer to 1,200.
That is awesome. I wish we could do that.
227.
Nice.
Guesses. Yeah, keep in mind each one of these samples can take from 15 minutes if we're finding like no scales at all, to sometimes it takes a couple hours to do one, especially with pellets since they have so many scales and other things that we count. A long time to do that.
Sometimes there's like 500 scales in one sample.
Yeah, and
it's
that's
pretty
a lot of
crazy.
clicking.
Yeah, we're over here in our corners just
Clicking.
Yeah.
That's good, lots of data.
Yeah.
Angela says 275.
Nice.
And then Brett asks, do you keep the samples for a long time? I wish you could see our storage up above us, our cabinets, where we have I'd say at least a dozen of these boxes filled with samples. And we do keep them because every now and then we'll have to go back and look at the data and see if that's really what we're finding. So sometimes we need just fresh eyes, years later to see if that was really what we think it was. And we experienced that recently. They were going through data from 2017, and we had to pull out all the ones that had rockfish in it just to see if that was really what we were finding. Because some of these colonies aren't known to have rockfish, for example.
So we keep every little sample we do.
Yeah, Meredith says we've kept all the samples going back to 2002.
Yeah, pretty crazy.
Yeah, but I find that really cool that we have the capacity to do that. And then also we use alcohol so that kind of preserves them. The alcohol will evaporate and dry off, and usually the scales stay intact, so they don't need to be like stored in jars or anything, which is good. But
yeah.
Okay, so we'll give you the final count, and
we still have more to do. We're almost done with this project for this year. Olivia and I have been doing this, and the grand total. Dun, 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 dun. 332 samples. So
I think Michelle
was the closest.
Very good.
Good job, Michelle. Yeah, 332 samples since June.
We still have about 40 more to go.
It's a really exciting project. We're gonna miss it when it's over. But we have a lot of other
we have lots of other projects to do. We have lots of samples coming off the Farallon Islands, which we'll talk about in our next live stream, like the Brandt's cormorant pellets, which is kind of the continuation of these pellets, but these birds are like way bigger and they have huge pellets. So they're similar to like the size of owl pellets, if you guys have seen those. So that will be exciting.
And they're surveying a different location too, so we're gonna get some different fish.
And this one's island fish instead of these coastal communities. So are these surprising rolls?
Yeah, Michelle, you get a high five.
Yeah, you get a sanitized high five.
So I think we're at our time now. Thank you for tuning in today. Hope you guys learned something new, enjoyed this during your lunch break or whatever you guys are doing. Feel free to send this to anyone who wants to know more about marine food webs and how we study the health of our oceans. In the next live, we will be talking about the Brandt's cormorant pellets. That'll probably be sometime next month. So look out for our posts about that.
And if you want to help continue to support marine science studies like this, our lab relies on donations from people like you. So feel free to check out the link that's in the description. Let's see.
There's the link.
There's a link. So feel free. Anything helps, thank you for supporting and even watching and sharing this video. It'd be great to get more supporters for Point Blue.
All right, enjoy the rest of your day.
Bye. See you next time.